All right, and also if for some reason I'm not talking loud enough or I'm not clear, just start yelling at me to speak up. Sometimes I mumble. All right, so if you're in the right place, this should look familiar. This is workshop 7, CHiP-Seq. So the outline for the workshop is on today, day 1. We're gonna mostly talk about principles and how to acquire data. We'll talk about some background, such as the assay protocol, principles and the theory, some quality control metrics you can use. We're going to download data from Geo using the SRA toolkit. And then we're going to end after we do alignment to the human genome using the Bowtie 2 sequence aligner program. And tomorrow it'll be more visualization of the data. We'll do the peak calling and we'll look at the data in the genome browser. We'll do differential peak calling. And on the third day, on Thursday, we'll do more downstream functional analyses. We'll look at CHIP region annotations using CAS. We'll look at how the peaks overlap with other data sets using CitePro and bed tools. We'll do some integrative analysis. We'll integrate RNA-seq data and look at promoters of differentially expressed genes. We'll do transcription factor motif discovery. And then we will assign peaks to genes and do gene ontology on the genes that are associated with the peaks that we're interested in. Okay, so chromatin is DNA and the proteins associated with it. So it's a complex of DNA and proteins. And CHIP is a way to study chromatin and specifically the locations of proteins that are associated with DNA. Coupled with sequencing, we can look at chromatin genome-wide and how chromatin is dynamically changing in different processes, such as in the disease states or during development. So this is a great way to study genome-wide organization of chromatin. And it's a very highly utilized assay because a lot of people want to study chromatin because it gives insight into gene regulation and mostly at the step of transcription. And if we study how chromatin is changing and how chromatin is being regulated in different stimuli, for example, you can get mechanistic insight into why gene expression is changing. So a complex, but probably even oversimplified model of how transcription is being regulated by chromatin is diagrammed here. So you can have transcription factors binding near transcription start sites and promoter regions, which can recruit general transcription factors, which will recruit RNA polymerase, also known as the pre-initiation complex, to orient polymerase to transcribe gene. And promoters are often marked by certain histone specifically H3K4 trimethylation is found near transcription start sites of active genes. And in gene bodies, you can have H3K36 trimethylation. And also H3 acetylation is often found near active transcription start sites. And also transcription factors can be found tens of or even hundreds of kilobases away from genes that they are regulating in distal androgenic regions or in a lot of times in introns, where transcription factors are binding these distal regulatory elements, also known as enhancers. And these can recruit cofactors such as chromatin remodelers or histone modifying enzymes. And these enhancers can be put in close proximity to the promoters they regulate through looping, and these loops can be stabilized through cohesin complexes or mediator complexes to aid in recruitment and stimulate pre-initiation complex to regulate genes that are very distal from these functional elements. So by using CHiP-Seq, we can study these factors that are important and study this highly orchestrated process in activating transcription. So I think it's important to know how the assay is performed if we want to interpret our data and know how to process the sequencing data. So the typical protocol involves cross-linking cells with a cross-linking reagent. Typically it's 1% formaldehyde. So that's to capture protein DNA interactions and also to prevent the loss of these interactions upon fragmentation of chromatin. So typically you fractionate chromatin with sonication, and you want to get DNA fragments to a size around hundreds of base pairs. And that way you can immunoprecipitate using an antibody that is specific to the protein or the factor, the histomodification that you want to study. And then that way you can enrich for DNA fragments that are associated with this antigen that you want to look at. So then you want to purify the DNA fragments. First you want to reverse cross-link, and then you phenylchloroform extract and ethanol precipitate the DNA fragments. And from this, you want to have about 1 nanogram of CHIP DNA, and it's typically measured by qubit, which uses a dye that binds DNA, and use that CHIP DNA to construct libraries. So you want to repair the ends of these DNA fragments, which will have random overhangs from the sonication. You want to fill in those overhangs and phosphorylate the ends and also A-tail the ends. So you have a phosphorylated 5' end and an A-tailed 3' end. So this allows you to have the proper substrate for adapter ligation. So the adapters are ligated, which contain for example, barcode or index sequences. So you can pool your libraries together and sequence them together and then demultiplex following sequencing. So once you ligate your adapters, you want to amplify these adapters with PCR. And then you have purified libraries that you will pool. So you have multiplex sequencing, which is a cost-effective way to sequence multiple samples. Then you can demultiplex these libraries using the unique barcode sequences. And then you generate files known as FASTQ files, which can be used for used by alignment programs to assign these fragments to specific regions of the genome using genome mapping. So typically you will do short sequences, short sequence reads. Typically now it's about 50 to 100 base pairs. And then you take those sequences and you want to assign them to a region of the genome using a sequence aligner, which is known as genome mapping. And using that, you assign these reads to a reference genome. And then you have an aligned file, a SAM or a BAM file. And then you can use these files to look for regions that are differentially represented in your chip versus a control, usually an input, and look for sites that are statistically enriched following your chip. And these are known as peaks. So now you have regions of the genome that you have putative, for example, putative transcription factor binding sites, or sites that have a specific histomodification, then you can do downstream analyses with this list of coordinates. And you also want to visualize the chip signal in a pile-up file where it shows the pile-up of reads, so you can visualize what the peaks look like in different regions of the genome. So you can create files such as wiggle files, which will bin the reads into specific steps, which will help to compress the file so it's not too large depending on the resolution you want. And you can use these for different programs such as genome browser viewing and other downstream analyses. So as I mentioned, peaks are significant enrichments of your chip over a control, usually an input. And what an input is it's a sample of your cross-linked sonicated chromatin that has not undergone immunoprecipitation. So this is essentially a negative control for sites that are enriched, or a negative control for peaks. And what input does is it controls for false positives. And how or what input normalizes for includes sonication bias. So it's known that certain chromatin regions sonicate better than others, specifically sites where the chromatin is more accessible or more open, as opposed to sites where the chromatin is more condensed, those sites are less sensitive to sonication. So open chromatin sites can often be overrepresented just by sonication bias. Input also controls for PCR biases during library amplification. So we know that there are certain DNA sequences that are preferentially amplified. However, those sequences will also be preferentially amplified in your input DNA. So input will control for those genomic sites. It's also controlling for sequencing biases. So certain fragment sizes, for example, can be have preference for sequencing. You can also have mapping biases during your alignment of your FASTQ files. So certain regions of the genome don't map as well as others, such as repetitive elements of the genome. So those sites will also be represented similarly in your input. And also a lot of transformed cell lines will have variations in genome copy numbers. So since we're taking the same chromatin sample for the input as with the chip, your input will control for genome copy number variations. So certain regions of the genome can be overrepresented if they are amplified, for example. So input, however, does not normalize for everything. It will not be able to normalize for antibody cross-reactivity. So if your antibody is not very specific and it's immunoprecipitating other factors, giving you false positives, the input will not be able to normalize for that. And also cross-linking artifacts. So if your protein is cross-linking to sites that it's not specifically bound to, there's no way to normalize for that from your input. All right. So here's a nice visualization of why an input control can help eliminate false positives. So for example, in this chip for tagged SCC1, I think it's a component of cohesin. So you see sites, I think this is in it, I'm not sure what, maybe this is yeast. You see sites on this region of the genome that seem to be overrepresented from your chip. So these are potentially peaks. However, when you normalize to an input control, you see that this site, which looks like it would be a peak from your CHIP sample, also is enriched in your input. So when you normalize against input, that'll eliminate that false positive peak, while the other peaks may be considered as statistically significant over your input control. So peak enrichment depends on how many reads you have in those specific genomic locations. So for example, let's say you have a site in your CHIP DNA, which you have 15 reads aligning there. So you have something that looks like a peak compared to neighboring sites. And in your control, if you have 10 reads also in that site, you have an enrichment of 1.5-fold. Now, due to the small number of reads, this is not likely to be considered a statistically significant peak, as it's only 1.5-fold enriched and you only have 15 or 10 reads aligning there. But sites that are more likely to be called statistically significant for binding, such as this site, which has 20 reads versus 5 in the control. So this is a fold enrichment of 4. Again, low number, but you have a greater fold enrichment. Well, on the other hand, if you have, again, 1.5-fold enrichment, like we did at this not statistically significant peak, this same fold enrichment is more likely to be called significant if you have more reads aligning there, because you have a higher N, you have more statistical power. So this alludes to is that the number of peaks that are being called from your chip is highly dependent on your sequencing depth. So, and this is in fact the case. So this is demonstrated in this graph where you see the fraction of called peaks versus sequencing depth. So as you get more mapped reads, so found on the x-axis is number of mapped reads in millions, the higher fraction of total peaks called increases. So this is displaying the number or the fraction of peaks versus millions of mapped reads for these different factors that were chipped in these different cell lines. So in all cases, you see as the sequencing depth increases, the number of peaks increase. So, and you would think that eventually it would level out, but for one of the samples, they went up to even 100 million mapped reads, which is typically much more than you would need. And the curve is flattening a little, but it's not completely flat. So it could, who knows how many more peaks you can call with increased sequencing. So that's why it's not exactly, the number of peaks is not really biologically relevant. So it's more of based on a technical parameter. It's based on sequencing depth. So it's not really valid to say that this factor binds the genome in this many locations, because it really depends on how much sequencing you perform.
I have a quick question.
Okay.
So does that mean that when you're comparing samples, say your control versus your whatever you chip, the read depth between those two samples should be similar? Otherwise, it would be difficult to compare when you're calling peaks after normalizing.
Yeah, so that's a great question. Most peak calling programs will normalize for that internally. So it's good to have as much sequence as you can, obviously. But if for example you have twice as many mapped reads in your input, they'll downsample the number of mapped reads in the input to match the chip.
Gotcha.
So it's not masking enriched sites. Does that make sense?
Yeah, thank you.
Yeah. All right, so as I mentioned before, peak locations can be used for functional downstream analyses. So we want to, once we execute our experiment, do our sequencing and alignment, we call our peaks, and the peak locations can be used for downstream analyses such as annotations. So you can see what features of the genome are enriched for where your peaks are located. Are they enriched near promoters? Are they enriched near distal energetic regions, etc.? Differential representation, usually if you have a experimental variable, how does that affect binding of your of the factor that you're chipping for? Also motif identification, usually transcription factor motif. I see there's a question in the chat. Can increase read depth potentially to increase risk for false positives / cross-linking artifacts? So read depth usually isn't related to cross-linking artifacts. In terms of risk for false positives, I think it would be the opposite, where the more read depth you have, I think it would eliminate false positives, because you are more likely to have a more accurate representation of the genome if you are if you have more reads. So you'll have, the background will look flatter, your input will look flatter. Where you have random variations is where you're more likely to have false positives called. I hope that answered the question. Also integrative analysis. So we can integrate other types of data sets, such as RNA-seq data sets or other CHIP data sets, is something we'll be doing later on, I think, day 2 or day 3. So it's also important to know CHIP-Seq limitations. So CHIP-Seq tells us putative transcription factor or cofactor or histone modification sites, but it's important not to overinterpret the data. So something you cannot do is compare data between different antibodies. So antibodies will have different efficiencies and different affinities for whatever factor they're raised against. So it's not really valid to do direct comparisons between antibodies. CHIP-Seq can't really tell you where a factor does not bind. So that would lead to false negatives. So I'll show an example of that here. So for example, this is a chip for a cofactor called YAP. So this is a standard single cross-linking protocol. So when you look at this track, the bottom one in blue, it looks like there are a few binding sites in this region of the genome. However, if you tweak the protocol to do double cross-linking, so oftentimes you get, for proteins that don't directly interact with DNA, you can increase your signal by doing an extra cross-linking step. You see, by this protocol, there are something like 10 times more peaks in this region of the genome. So if you say that the factor doesn't bind based on the single cross-linking chip, you could be creating false negatives just based on technical issue. So one thing you could argue is that due to double cross-linking, these could be cross-linking artifacts, and these could be actually not real binding sites. But in fact, at most of these peaks, we see the transcription factor motif we would expect YAP to bind at. So these are probably, in fact, bona fide YAP binding sites. So that's why I say it's you know, be careful when calling sites that don't bind. You could be potentially incorrectly calling false negatives. So the data is correlative as opposed to functional information. So not all binding events or putative binding events are necessarily functional. You have to do different experiments to determine that. And also the data comes from a cell population. So interpreting binding on a cell-by-cell basis is not really possible from a bulk cell population, which is necessary for CHiP-Seq. So we don't know exactly what the peak height signifies. It could be that a higher fraction of the cells have the binding in those sites, or it could have higher affinity in a small fraction of the cells. So I'll talk a little bit about procedure troubleshooting. So more often than not, if you have poor enrichment, it's usually due to an antibody that's not very suitable for what you want to chip. So that's usually the go-to for troubleshooting. So what I recommend is try to start with an antibody that's either chip grade. So a lot of commercial antibodies have been tested for chip, such as cell signaling. They signify here if the antibody works for chip. Another good resource to know if your antibody is likely to work is just to look through the literature and see what other people used when they chipped the factor that you're interested in. But be careful because a lot of antibodies are polyclonal, and typically how well they work for chip can be lot dependent. So it's better to, if you're going to look through the literature and use an antibody that was published to work, it's better to use a monoclonal, so you know you're getting the same exact antibody that they used. Another issue that could lead to loss of signal is oversonication. So oversonication can lead to loss of protein DNA interactions. And this can be due to just the physical force of the sonication, breaking apart those interactions. And some proteins are actually sensitive to protein degradation from the sonication process, such as RNA polymerase, actually, which is a common chipped factor. So that protein can start to be degraded from sonication, especially in the presence of SDS. So here, this is showing fragment distribution following different sonication protocols. And so you can see as you increase the time of sonication, you get smaller and smaller DNA fragments. So what I would say, I would stay clear of sonicating down to this level where you have pretty much all of your DNA below 500 base pairs. So depending on what you're chipping, this could be over-sonication. I tend to stay on the lighter sonication side, so this would probably be ideal. I would even use the second sonication protocol depending on what I'm chipping. So if you're chipping histones, you can sonicate the crap out of it, and the histones will still be there because there's so many contacts between DNA and histones. So it's really hard to over-sonicate with histones. But for proteins that have less contacts or don't bind DNA directly, such as co-activators, I would stay more on the lighter sonication protocol. Another thing you can do, as I mentioned earlier, is double cross-linking. So a typical protocol would involve using DSG and formaldehyde. And I've had luck with improving signal, especially with proteins that don't bind DNA directly, for increasing signal with double
Nathan,
cross-linking.
a quick question.
Yeah.
What is your opinion on, so I know that there's protocols that use MNase as a means of kind of digesting the DNA. Would you expect treatment with, or some combination of sonication and MNase to be some workaround for the oversonication issues, or for sensitive proteins like RNA polymerase or others?
Yeah, so I don't have any experience using MNase. So are you referring to it as an alternative to cross? Because there's something called native chip where you don't cross-link and you use MNase, or cross-linking and then MNase digestion.
Yeah, I was suggesting cross-linking and MNase digestion. I've actually done that before with RNA polymerase II chips,
Okay.
but I wanted to hear your perspective.
Yeah, I mean, I actually, I don't really have a lot of experience with that. I think you're pretty limited as to what, yeah, I'm honestly, I don't have a lot of perspective on that because I haven't done it.
Okay.
But
That's cool.
my understanding is that it won't work for every factor that you're, like a transcription factor, it might digest away too much of the DNA, like a small factor, and so you're going to get fragments that are too small to map. So for histones, I think it works great, but with histones, cross-linking or sonication is never an issue anyway. And Pol2, as I mentioned, it can be degraded from sonication, so I see that as a workaround. And apparently, I mean, you say you've had experience with that, so that's good to hear.
Yeah. Cool.
Yeah. All right. So as we know, sequencing costs money. So a good way to prevent sequencing chips that aren't going to give you valuable information and making your PI mad is to confirm that the chip worked prior to submitting your libraries for sequencing. So to do that, you really need to have a site in mind that you expect your defined enrichment at. So, which isn't always the case, but a lot of the time a gene that is expected to have enrichment for the protein you're chipping, or for example, this was a chip for acetylation of H3K18, which is known to be promoters of active genes. So with this chip, I did qPCR prior to library construction. So I used primers designed at the cyclin E2 promoter. And by doing this, I confirmed that I had enrichment in the IPs, or the chip samples, over inputs. So that gave me confidence that the chip did in fact work. So I could submit this for sequencing libraries. Otherwise, something you could do is maybe somebody else is sequencing a library. You could ask if you could put in a small sample of one of your libraries and just get a little bit of sequence back for your chip, and see if it looked like it worked. And if it did, then you can go ahead and sequence the rest of your libraries, or sequence more of your libraries. So an alternative, or a couple alternatives to CHiP-Seq that have been published more recently, are cut and run, and even more recently, cut and tag. So these protocols are designed to give you similar information as CHiP-Seq. It can tell you specific histone and transcription factor binding genome-wide, but it has some modifications that might be more appealing to you, depending on what you want to, what factor you're interested in. So the cut and run protocol involves isolating nuclei, but actually I think updated protocols skips the nuclear isolation and uses cells that are permeabilized, maybe by digitonin, so which allows you to diffuse in your antibody. So you're still using an antibody that's specific to, say, a transcription factor. And what this protocol uses is protein A fusion to MNase. So protein A will recognize the antibody, and then the MNase is an endonuclease, which will cleave the DNA. So you want to increase the local concentration of this MNase to regions that are near your transcription factor, and then you treat, or you diffuse in calcium to activate the enzyme. So this will cleave the DNA around the transcription factor, and then this transcription factor antibody MNase complex will then be small enough to diffuse out of the nuclei. And then you can take these small DNA fragments to construct libraries. And this avoids cross-linking, so you don't need to worry about cross-linking artifacts. You can computationally remove background. So by throwing out, if you do paired-end sequencing, you can throw out fragments that are larger than a certain fragment size to remove background. And because of this, it's reported that you need about 1/10 of the sequencing depth that you would need for CHiP. And what I think the best aspect of this protocol against CHiP is it requires a very much smaller amount of cells, 100 for histones for cut and run, to thousands for transcription factors. But more recently, this updated protocol, cut and tag, it's been reported to been able to work with single cell. So this is essentially single cell CHiP. So the cut and tag protocol, which I think is very clever, but you're still using an antibody, so you're still limited by the efficiency of your antibody. So again, you permeabilize the cells, diffuse in your antibody and a secondary antibody. And the reason for that is to get even higher local concentration of the protein A. And now the protein A is fused to the TN5 transposase, which is the same enzyme used in ATAC-seq, which will recognize your antibodies. And again, I think maybe magnesium is used to activate the enzyme, which will then cleave the DNA surrounding your protein of interest, and it'll ligate sequencing adapters, which are tagmented to the TN5 transposase. So this is an improved protocol versus the MNase, specifically because it seems to be more robust than cut and run, and you save yourself the time needed to construct libraries because you are directly ligating your sequencing adapters by the TN5 transposase. So I think that's going to be an exciting assay as it gets developed more. So I will say, before you go out and switch over to cut and run, these assays are at least cut and run, which I've used, and people I know who have tried it have only gotten it to work with histones. It seems to be pretty tricky with non-histone proteins, which histones aren't really a problem with CHiP-Seq anyway. So I think it maybe the cut and tag will be the answer for your CHIP troubleshooting, but we'll see. This is still pretty new. It's just published last year. So now I'm going to go into some quality control measures. So I just want to say before going through these that I don't think that these are necessary to do every time you do CHiP-Seq. I think this is more of a, if your chip didn't work and you don't know why, these are certain tools that you can have to try to determine what went wrong, or how to best approach the assay when you redo it. So, as I said, I don't recommend you just starting with all of these quality control measures. But anyway, it's good to know these tools in case they are they could come in handy in case you need them. So library complexity is often an issue when getting high quality data. So library complexity is determined by the fraction of DNA fragments that are non-redundant. So when you have low complexity libraries, you have a high fraction of clonal reads. So just to visualize that, here, a typical CHiP-Seq will have this pile up of reads in this, this distribution. And as you can see, none of the reads are identical. They are all aligning to unique positions. While when you have low complexity libraries, you have a higher percentage of clonal reads, and clonal reads will look like this, where you have the same exact read aligning in the same position. So you have multiple copies of the same aligned read, and that will result in low quality data. And sources of low complexity libraries can be from a low DNA recovery from your IP. So if you are inputting a low amount of DNA into your library construction, then those fragments are going to be overrepresented, and you're going to have the same fragments seen over and over when you amplify with PCR, or when you sequence the same fragments multiple times. PCR bias, of course, sequencing bias, if you had the same fragments being over-amplified or over-sequenced, and adapter dimers. If you have the incorrect molar ratio of adapter dimers to fragments, then the adapters will ligate, and then you'll have too many of those contaminating your libraries. So something you can do to assess the library complexity is look at the non-redundant fraction. So this is the number of distinct uniquely mapped reads, typically after removing duplicates, over the total number of reads. So by ENCODE standards, if you have less than half of your reads being aligning to that are non-redundant, that is what they would say is concerning. If you have between 0.5, half, and 80% being non-redundant, then that's acceptable by their standards. Between 0.8 and 0.9 is compliant. Greater than 0.9 is ideal. And you can get this number after doing alignment, where it'll report how many or what percentage of your reads aligned exactly one time in the genome. And so typically like this, you should be getting greater than 90% in a high quality data set. So this program called Homer, which is a suite of CHiP-Seq tools, will report the number of reads per position, and the fraction of total reads. So in an ideal CHiP-Seq experiment, you want most positions only having one aligned read, right? So around 90% of the reads should be aligning to one position. Non-ideal, you'll have less than 50% of the reads per position. So this is a way to visualize that using this clonal tag distribution plots. So if you have low DNA recovered from your chip, when you do the adapter ligation, you're going to have the incorrect adapter ratio, which normally you want the fragment to adapter ratio that'll allow for all the adapters to be ligated to fragments. When you have a low input into your library construction, you have a higher ratio of adapters, which will lead to a higher percentage of adapter dimers contaminating your libraries. And when you visualize the distribution of these fragments of your libraries on a tape station, for example, if you have contaminating adapters, you will see a peak around, I think like 120, depending on your adapter size. And the lower the amount of DNA used for your adapter library construction, the higher the number of adapters, as shown here. 10 picograms is a very small amount of DNA. So this is 2% having adapter dimers, 100 picograms, you still can see a peak. 1 nanogram of chip DNA used to construct libraries. Hopefully you'll get 0% adapter dimers when assessing your fragments on the fragment size on the tape station. So there are methods to clean up your libraries to remove adapter dimers. And we will have breaks. Let's see, it's been about an hour. So I think, why don't we take a 5-minute break right now? So we will return in 5 minutes. So it's a good time for a coffee break or bathroom break, or doing a somersault on your couch, whatever you need to do. So I'll see you guys in 5 minutes. Okay. So we were talking about adapter dimers and how they can contaminate libraries to cause low complexity. And by looking at the non-redundant fraction of reads in your returned from your sequencing, you can assess the complexity of your libraries. So typically you do short sequencing reads on the ends of your fragments. So while your fragments following sonication and library preparation, they're usually hundreds of base pairs. However, if you're doing 50 base pair reads when you submit these libraries for sequencing, you're typically sequencing the ends of these fragments. So if you're doing paired end sequencing, you know the exact fragment length, but if you're doing single end sequencing, which is all you need for CHiP-Seq, what you'll get is a theoretical pile up of the reads that are to the left of your binding site for the positive strand, for the reads that align to the positive strand, and to the right of the binding site on the negative strand for the reads that align to the negative strand. And when you look at the distribution of these tags, the distribution for each strand will look like this, where the the estimated fragment size is the distance between the top of this curve and the top of this curve, if you're sequencing both ends of the fragments, and the theoretical binding site for your protein of interest is directly in between these two curves. So using these distribution of tags, you can more accurately predict the true binding site, which, as I mentioned, is directly in between these two curves. So some programs will do modeling to predict where the more accurate binding site is, but you can use this sequence distribution to assess enrichment, to assess how well your chip worked. So for example, you can look at what these different statistics that this program Homer uses. So these are same strand fold enrichment. So this is where you look at so the same strand and different strand fold enrichment. So the density of B based on this plot is the density of the reads that are piling up here. So if you have a lot of reads that are aligning in peaks, you'll have a distribution of reads like this for the positive strand, and a certain distance away for the reads that are piling up on the opposite strand. So by looking at the density of these reads in this region over the region just outside of these, you can get an estimate of how much enrichment you got for your chip, how well your chip worked by calculating the density of B over the density of A and C. Also, you can look at the same over different strand fold enrichment, where you divide the density of the positive strand for B over the density of the opposite strand for in this section B. So these should be about equal if you have good chip enrichment. So using this different strand enrichment, you can do cross-correlation analysis, where as you shift one strand towards the other, k number of base pairs, you will get the highest correlation when the reads are directly, where the peaks of these two distribution of reads are perfectly aligned. So at the point where the highest correlation value occurs is your theoretical fragment size for how many base pairs you shifted the distribution of the reads from the opposite strand. So when you plot the cross-correlation values, so this is similar to like a Pearson correlation value over the number of base pairs of strand shift. At the top, the highest cross-correlation value is at the strand shift for the value of your estimated fragment size. So of course the fragments are going to be different lengths, but this is the length for the average fragment size. And you also get something called a phantom peak. So you get this peak, a very sharp peak, which is due to the read length. So this peak will peak exactly at where the sequencing read length was. So if you have 50 base pair reads, you'll get a phantom peak at 50 base pairs. So you'll get a sharp drop off past 50 base pairs. So the CHIP peak equals the predominant DNA fragment length as you shift these strands, so the highest cross-correlation value, and the phantom peak equals the read length. So using these values for cross-correlation, you can also assess the quality of your chip enrichment. So for example, ENCODE will calculate the normalized strand coefficient. So this is the cross-correlation value at the fragment length. So that's here. The CHiP-Seq fragment length is, again, where the cross-correlation is at the highest value as you shift the reads from one strand towards the other. And divide that by the minimum cross-correlation value. So that gives you an NSC, a normalized strand coefficient. So to pass ENCODE standards, that value has to be greater than 1.05. And also you can calculate a relative strand coefficient. So this is the cross-correlation fragment length, again, the estimated fragment length cross-correlation value, minus the minimum cross-correlation, over the read length cross-correlation minus this, so that for that value, if it's less than 0.8, then ENCODE would repeat the experiment. So here's an example. This is a very good enrichment. This is marginal. As you can see, the fragment length peak is very low compared to the read length, and failed would be you don't really see much of the fragment length. So a more simple metric is the fraction of reads in peaks, or the FRIP. So how many reads were found in peaks or sites of significant enrichment. So it's typically, if you have close to 10% of your reads in peaks, that's a very, I would say that's a high quality data set. But of course, as you would imagine, it really depends on what factors you're chipping. Some factors aren't as widespread in binding the genome. So you're gonna intrinsically gonna have less peaks, and the FRIP is gonna be lower. So take these metrics with a grain of salt, I would say. But one thing that proves that it is somewhat useful is that the higher the normalized strand cross-correlation value seems to correlate with a better FRIP value. But again, the best way to assess, I think personally, the best way to assess if your data set is going to be useful to you is just by looking at the data. So genome browser inspection, I think, is just really going to give you the most information, because there's not really set values that are standard for any chip you're going to do. I mean, you're going to get a wide range of expected values depending on what you're chipping. But if you look at what the data looks like in the genome browser, you can get, it's not an objective way to assess data quality, but it really depends on what you want to get from the data. So what you can see is this experiment, this replicate for EGR1, it looks like you have low enrichment compared to background at these different promoters. This replicate looks a little better. And here these were redone, and you can see that the enrichment is a lot better. So these data sets will probably give you more peaks and probably give you a more accurate representation of what this factor binding looks like in the genome. But really, I mean, it's not that this replicate isn't going to tell you, isn't going to be useful to you. It could be. I mean, you can see traces of binding. So really, if that's all you got, don't just throw it away. Try to be resourceful. Another metric for this time to assess reproducibility between replicates is what's known as the irreproducibility discovery rate. So this is sort of analogous to a false discovery rate to assess how reproducible your data was between replicates. So if you have replicates, you can you can obtain a high confidence peak list by only using the peaks that were giving you an IDR value of less than 1% or 0.01. So as you can see, these are going to be the peaks that have the best signal. So this is plotting the log signal for one replicate versus the log signal for a different replicate. And the peaks that passed an IDR of less than 1% are going to be the peaks that give you the better signal, which makes sense. And this is also demonstrated here by, instead of looking at the log signal, the peak rank, with the lower the rank number, the better the signal was. So the smaller the rank, those are going to be the high confidence peak list. And as you relax your criteria for calling peaks, the peaks with a higher IDR, so the less confident list, is going to increase. So you're going to have more peaks, but these peaks are going to be less consistent between replicates. So some other calculations you can do in terms of assessing reproducibility between replicates, again, is calculating the IDR between replicates to get a number of peaks that are consistent between true replicates. This is going to be an NT, a value that's represented by NT for the true replicates. And then you can compare that to an NP, which is a number of pseudo replicates. So to do that, you pool the two replicates into one, and then randomly separate those reads into two pseudo replicates, and then calculate the IDR between the peaks and the pseudo replicates. And that's going to give you the number of peaks consistent between pooled pseudo replicates, the MP. And then if you divide those numbers, the MP over the NT, you want to get less than 2, otherwise that's considered low reproducibility by ENCODE standards. And another calculation you can do is N1 over N2. So that is when you take each replicate and divide each replicate into 2 pseudo replicates. So now you have 4 pseudo replicates. And then you take replicate 1 pseudo replicates, calculate the number of consistent peaks with the IDR value, that's your N1, calculate the consistent peaks from the 2 pseudo replicates of replicate 2, that's your N2, and divide those. And if that number is greater or equal to 2, again, that would be considered low reproducibility. And then you can check those replicates for other QC measures, the NSC and the RSC, which we talked about, FRIP, NRF, to look at if the sequencing depth gave you adequate number of mapped reads. The read quality also can give you poor data. So if you're interested in this, here's some examples of programs to look at these quality control measures. So we aren't going to be using these programs for the interest of time, but if you're interested, there's some links. So here is a schematic of our pipeline. So what we are going to do is we are going to download data from which are stored as SRA files from GEO. It's the Gene Expression Omnibus. So it's an NCBI curated database. So we are going to download files, convert them to FASTQ files. So these contain raw sequencing reads. We are going to use the FASTQ files to do alignment to a reference genome and generate SAM-BAM files. We're going to do peak calling using the program MAX, which are going to generate two two main files, bed files and bed graph files, which can be, bed graph can be converted to a similar file, wiggle files, and compressed binary bigwig files. So these are pile-up files which have more information than bed files. Bed files are basically the peak locations. And using these files, we're going to do downstream analyses with various programs to look at intersecting peaks, gene region annotations, transcription factor motifs, gene ontology. We're going to look at the files in Genome Browser, do promoter analysis of regulated genes, and visualize signal at regions of interest. So here is a model that we are going to test using CHiP-Seq data. So this is the interferon pathway, which involves these secreted interferon alpha or beta molecules, which bind to interferon, cell surface interferon receptors. So these are phosphotyrosine receptors, which will heterodimerize to activate a kinase cascade, which will lead to phosphorylation of cytoplasmically restricted transcription factors. So upon their phosphorylation, they will form these complexes, which will then allow them to translocate into the nucleus and bind to certain DNA sequences, which are known as, for example, the interferon stimulated response element. So this complex known as the ISGF3, so we're going to focus on this complex, which is composed of STAT1 and STAT2 and IRF9. So upon phosphorylation of STAT1 and STAT2, they will form this complex with IRF9. It's believed that IRF9 is the direct, well, actually, I think STAT1 and STAT2 also make contacts with the DNA. But the model is that this complex, upon interferon stimulation of cells, will form, translocate to the nucleus, bind to specific DNA sequence elements that are near the promoters of antiviral response genes. So upon binding these promoters, these genes will be transcriptionally activated, and it'll express genes that will form products that are known to help protect the cells from pathogen and viral infection. So, so just based on this model, we are going to test some hypotheses. So we are going to test the hypothesis that STAT1 and STAT2 bind the same genomic region since they are in the same complex. We are going to test STAT1 is found to bind ISREs upon interferon stimulation. And we are also going to test STAT1 binding is near promoters of interferon alpha activated genes that are enriched for antiviral and inflammatory response. So to do this, we are going to obtain data that was generated by this ENCODE consortium. So this, I kept referring to them for the quality control measures, but this is a international effort to make available publicly available comprehensive lists of functional elements in the human and also mouse genome by generating high throughput sequencing data such as CHiP-Seq data. So we're going to utilize this, the data that ENCODE generated, which has been you can, I think you can download it from both the ENCODE website, but also through the NCBI database, GEO, the Gene Expression Omnibus, where researchers will upload their data sets to be publicly available to researchers. Usually it's required for them to upload the data sets upon publication. Most journals will require you to upload this data. So we are going to download these CHiP-Seq data sets in K562 cells, where these cells were treated with interferon alpha for 6 hours, and then STAT1 CHiP-Seq was performed, and interferon alpha treatment for 30 minutes, followed by STAT1 CHiP-Seq. So we can download the raw data through the sequence read archive, where the data is stored as SRA files. We can download this data using the SRA toolkit, which we will download and then run from the Hoffman server. So in order to participate, everybody's going to need a Hoffman account, which should have been required for you to register for this account. So I tried to make this very easy. I tried to design this workshop for people who have little or almost no computational background. So to do that, I made it so, well, I tried to anyway, make it so you can directly copy and paste the commands from the PDF I emailed from the slides. So if you want practice typing them out, that's great. If you want to copy and paste them, that's fine too, whatever you want to do. But be aware that regions where there's red text, you're going to have to alter the command. So for example, this first command that I put to log into your Hoffman server, you're going to need to replace your username with your actual username. So what we're going to do right now is log into Hoffman. Of course, you need to use your password. Hopefully everybody knows their password. So what I like to do is work from the Flash Scratch folder, which is probably going to be necessary for this workshop. So in case you don't know, this is a folder that's given to every Hoffman user, which I believe you have 2 terabytes of storage. So this is the pathway to that folder. So of course, this isn't going to be your folder. This is my username. So to get to your Flash Scratch folder, it's going to be the first letter of your username followed by your username. But what I like to do is create a shortcut from the home directory. So after you log in, you can create a Flash Scratch shortcut by using this command. So when you use this command, make sure you don't have a space or after this. So this is going to name the symbolic link or the shortcut as Flash Scratch. And then we are going to request an interactive session using this command. So in a minute, I'm going to go through these commands on terminal. But in order to run jobs on the Hoffman, you need to request resources. And one way to do that is request an interactive session, which we'll be working from these workshops. So here's the command to request an interactive session. We're going to request 4 gigabytes of memory for 2 hours, which should be more time than we need to finish the workshop. And we're going to request 4 nodes. So the more nodes, the more memory, the more resources you're requesting, the more time also. So the more you request, the more it might take to be granted the interactive session, but I think this is a low enough number where it won't take us long to get this interactive session. Then we're going to enter the Flash Scratch. We're going to download the SRA toolkit from NCBI so we can download FASTQ files from those chips that I mentioned. So we have to extract this zipped folder. and then we will download the data from, download the raw reads from SRA to generate the FASTQ file. So if you go to NCBI, you can click here, you can see information about the data set that we're gonna download, how many spots or how many reads it has. And what we're gonna do is download both the CHIP FASTQ files for stat1, 30-minute, 6-hour interferon alpha treatment, and also the input. So we can use that as a control for peak calling for both. So we're gonna download and generate 4 FASTQ files. So we'll start with that. I'm gonna open and share terminal so I can type along with you. Let me know if the screen, so I'm trying to make it so you guys can see what I'm doing, but let me know if there's any issues with that. So I'm gonna log into Hoffman. You guys should do this too. Obviously, I'm not a very good typer. You won't see your password as you type it. And these are probably things you've covered from oh, sorry. So yeah, if you're gonna copy and paste the commands, don't include this $. Start the command. Sorry, I guess you can't see what I'm doing. Hold on one second. So if you're gonna copy and paste the command, whoops. I don't know what I just did. Don't include the $, or the space before the command, and try not to include the, what's going on? Started to email my username. Try not to copy anything after the command either, because that might confuse it. So do it like this. All right. I'll probably type out most of them. So I logged into Hoffman. I'm on my home directory. I'm going to create a flash scratch shortcut. Going to be named flash scratch. It's going to tell me that this file already exists because I already have one, but if you don't already have one, it won't say that. And then we are going to request an interactive session. Hopefully it won't take very long to do that. Though, no, it should be fine. You'll know when your session got granted because your login, your username will no longer say your username at login. It'll have some numbers, which is the session number that was designated. Okay. So when you request so again, any text in red, you need to replace to match your, specific to your username. So when you create the flash scratch shortcut, replace the Z in the XEM key with the first letter of your username and then your username.
Oh, gotcha. So the Z is not like a folder within which I'm trying
Well,
to create the
it is a
flash scratch.
folder. It is a folder, but your folder is different than my folder. So every Z user is put in the Z directory. But if you're not, if you're using
Gotcha. Okay.
then yours won't be in that directory.
All right. Thank you.
Yeah. Okay, so my interactive session was granted. So what we're gonna do next is enter the directory we're gonna work in, which is the flash scratch, cd flash scratch, and then we're gonna download the SRA toolkit, and this is a long link, so I'm gonna copy. Oops, I just realized they're hyperlinked, so be careful if you click on them. But I'm gonna copy this link in, so that should allow me to download this compressed folder with the SRA toolkit. Okay. Okay, so if you're having trouble making the symbolic link or the shortcut, make sure that there's no spaces after anything. Make sure make sure you have the slashes in the right places and no extra spaces. But if that doesn't work, and you're not able to get the shortcut created, you can enter your flash scratch without using the shortcut just by cd and then the directory where your flash scratch is found. So I would enter it like that. Okay. All right. So I downloaded the I see some people are still waiting for their interactive session to start. Has anybody gotten it to start? It took a few minutes for me. All right, well, for what we're doing, so the reason you want to request a interactive session is because you can get kicked off for using resources when, not kicked off, but you could have your job killed for using resources that that you didn't request. But we're not going to be using a lot of resources today. So I think it might be okay to, if you can't get the interactive session to start, just try doing the jobs without it. Even though I know that's not what you're supposed to do, but I think it'll be okay. I don't think your job will get killed. We're not doing too intense computational stuff. So make sure you don't copy the $ if you're running the command. You start after $ space, so you would start the command with ln space -s. Okay. So for the tar command, this is going to allow us to extract and unzip our SRA toolkit. So these options mean decompress or expand the toolkit and unzip it and display as it's being extracted. So there's a lot of files in here. It shouldn't take very long though, so it's already done. So if you're having trouble with the command, try copying and pasting it. Because oftentimes when I have trouble, I type something incorrectly. So now I have the SRA toolkit extracted, and I'm going to use this program that's inside of the folder. So in the folder, and then in the bin directory, there's this utility called FASTQ dump, which it will take the SRA file from Geo and it'll output it as a FASTQ file. So that's found on the page 36 of the slides. So SRA toolkit bin FASTQ dump minus capital Z. And then you want to type in the identifier for the specific data set that we are going to download from NCBI. And then we are outputting it as the name we want to give it, which is stat1 30 minute 30M IFNA interferon alpha dot FASTQ. And then so this, I've noticed that this is prone to having errors. So if it says that there was an error, you can stop it with, I think it's either Control Z or Control X, and just do it again. I know sometimes I have to do it 2 or 3 times before it works. I don't know why, but the connection gets interrupted a lot. But if that happens, just try it again. So if you're still waiting for the interactive session, just open up a new tab in Terminal with Command T and log in, and this time skip the request for an interactive session and just go into your Flash Scratch folder and download the SRA toolkit. And just start downloading the FASTQ file. And if you're not able to make the Flash Scratch shortcut, just enter the directory with CD and then the directory path, which is the part after the ln space -s. So each FASTQ file should take 5 to 10 minutes to generate. So this is a good time to stretch your legs, go to the bathroom, whatever you need to do. I see my first one finished. So I'm going to just press up, alter the name to 6 hours, stat one 6 hours, change the SRR link to the 6 hour one, which is SRR 502327. And it tells you how many reads were downloaded into the FASTQ file. So while we're waiting to generate our FASTQ files, I'll just explain a little better what FASTQ files are. So these are the files that you're going to get after you demultiplex your libraries based on barcodes. And if it's single-end reads, like what the CHiP-Seq dataset we're downloading, you'll have one FASTQ file per library. If you did paired end, you'll get two, and a read one and a read two. And what the FASTQ files contain is 4 lines per read. So the first line is a sequence identifier. So this isn't going to be very useful to us. It's just kind of some information about the sequencing run in the cluster. The next line is the actual sequence that was called. The third line has a separator, which is it's always a plus sign. I'm not sure why. And then the fourth line has information about the quality of the called bases. So this is known as a Fred score for each base that was called using this code, this ASC2 character code, which assigns a quality for each base that was called. If you want to learn more about that, you can Google the ASC2 Fred code. But basically, it tells us how confident each base was that was called. So I hope this is working for, I hope everybody can get this to work in some way. This is, like I said, this is the first time doing this strictly over Zoom, so it's harder to help people when I can't actually see their computers, but I hope this is working out. And the second one just finished for me, so I have 2 more. So I'm going to do the input 30-minute one. Okay. Oh yeah. One thing I should mention. So the Flash Scratch folder that we're working in, while it's pretty generous that they give us 2 terabytes of storage space to work with, which I you know, you can do a lot. I rarely fill it up. It does happen. The thing is, your files can be deleted after 14 days if they've been untouched for more than 14 days. So it's not a good directory for long-term storage. So hopefully you can use that folder to process files and then move them somewhere else. Maybe maybe your own lab has some storage space, or I know UCLA has given storage space on Box. Google Drive, I know, has free storage space. So just be aware that files will be deleted if not touched in more than 2 weeks. And one way to touch the files, if you want to keep your file there but you haven't been using it, so I'll type in the command. You can just use the touch command and then a star, which will touch everything in your, I just typed in the chat, touch star will touch all the files in your directory, so that will keep them safe for another 2 weeks. So if you remember to do that, you can store them in there. Something else, I know some of you are taking this workshop for course credit. I think Bioinformatics 275 or something like that. There will be 2 quizzes. We'll do a quiz at the end of the workshop today, and then there'll be another quiz at some point on Thursday. So I'll send the link to the quiz for every attendee, but it's only required if you're taking this workshop for a grade. Otherwise, feel free to ignore it. You can take it if you want, whatever you want to do. I'll let you know when I send out the quiz. It won't be, it'll be after we finish this. About the touch star command. So that'll only touch the files in the directory that you're in. So if you have deeper directories, if you have a directory within a directory, you can touch those files with touch star /star. So that'll touch all the files in second directories, and you can keep going with that until you've touched every file in every directory. So again, that's just a way to keep your files from being deleted in the Flash Scratch. And the deadline for the quiz today will be, that's a good question. I didn't think of that. How about a half an hour after I send it out? I might send it out while we're doing the bowtie 2 alignment, because again, it's sort of more dead air, so that might be a good time to do it. But it's only 5 questions, so and you can use your notes. So you have a half an hour to finish the quiz. All right, I'm on the last one. All right, while the last one is finishing, I will talk about what we're doing next. So, I hope this is showing up. We are going to align our FASTQ files to the human genome using the Bowtie2 program. So it's a sequence aligner. It's a so-called ultra-fast and memory efficient tool for aligning sequence reads to long reference sequences. So what Bowtie does is it takes each sequence, a DNA sequence, and it tries to assign it to the best match in the reference genome that we use for alignment. So they define alignment as the process in which we discover how and where the read sequences are similar to the reference sequence. So what it does is it determines scores for each potential reference sequence, where each potential alignment region. So if the read matches, well, down here you can see there's an example of a read and how it aligns to a reference sequence. So you can see that there's a mismatch here in A to T. There's an insertion of 2 G's here. So this insertion is going to penalize the score, and same with this mismatch. So for each region, each potential alignment region, it's going to calculate a score, and it's going to result in aligning the read to the reference site with the best score. So you can play with the parameters to have specific cutoffs of what scores are required for the alignment. Typically you want to have each read aligned to only one region of your reference, since it only came from one region of the genome. So if it scores similarly to, if it doesn't have a different score between 2 regions, for example, then we aren't going to assign the read to either region. That's the parameter we're going to use when we do the alignment, because we aren't confident as to which region the read aligns. We'd rather not have a false positive. So let's see if, yeah, this is still going. So as soon as that one's done, we will align each FASTQ files using Bowtie2, and I'll go into more. So I guess I'll just talk about it now. So so some of the parameters we're going to use are shown here. So if you just type in Bowtie2, it'll show different settings you can use. So we're going to use a Bowtie2 index file, which you designate with the -X option. Sorry, this is so small. The -Q option that we're putting into the command just tells it that we're inputting FASTQ file. We are going to use this -K option. We only want to report one alignment per read, and we are going to tell it not to keep the reads that didn't align in our output. So this suppresses reads that didn't align. So this will just help to reduce the file size for our SAM file that we're going to generate. In the -P, it's going to say how many threads, basically how many processors you use. So the more you use, the faster the alignment will go. This --local option Oftentimes you want to clip the ends of your reads, since the ends of the reads are more prone to have errors. So as you know, there's less accuracy in calling sequencing, base calling for the ends of sequence reads. So the minus local option will allow for mismatches at the ends, which is called another term they use is soft trimming or soft clipping. So instead of doing hard clips of all the reads, we're going to use the -- local option, which will take into account that the ends will be mismatches at the ends will be less penalized than mismatches in the middle of the reads. And we will download our reference genome from the Bowtie 2 website. We're going to use the HG19 reference genome, human genome 19. And this will be the commands. So let's see if mine finished. So mine all finished. So I'm going to start by using the commands on page 42 of the slides. So I'm going to download the HG19 reference genome from the Bowtie 2 website. So if yours aren't done yet, don't worry. You can do the alignment at any point once they finish. I'm just going to go ahead because time is getting low. It's 3:45, so we still have 45 minutes. So we should be done before then. But if you need to do the alignment still, then you should have time. So I'm downloading with the wget command, the HG19 index files. As long as I typed it wrong, but I'm getting a lot of errors. So maybe I did something wrong. Let's see. I can't figure it out. So I'm going to copy and paste it. Error. Well, I've never seen that before. Has anybody tried this yet? If so, let me know if you got it to work. Okay. And there's something wrong on my end. I've never seen this happen. All right. Here's what I'm gonna do. I already have them downloaded, so actually maybe I don't. Sorry, I will make these available to anybody who's also getting this download error. Oh, to stop the error message, hit Control Z. It should kill it. So if you're like me and you're having trouble downloading the HG19 reference from that link, I'm going to put up a command. Actually, it's going to be a few commands to use to copy them from my folder, because not really sure why that error is happening. So hold on 1 second. How many people are also getting that error when downloading the hg19.zip folder? Okay. So there's going to be 6 files that you're going to need to copy to your Flash Scratch folder. And I'm going to paste the command for you guys to use so you can copy it from my folder. Sorry about that. I tested that command yesterday and it worked, so I don't know what the connection problem is.
And I ran it a little bit before we got to it, and it worked.
All right. Hopefully you can see this. There's these 6 files that you need. So you'll just use the CP or copy command to, from my folder, this is the file, to space period, which as long as you're in the Flash Scratch folder, will copy to your Flash Scratch. And these are the Bowtie 2 reference files we'll need. And they're a little large, so each one will take like 5 seconds or so. All right. I will also copy and paste them in the chat. So if you're having trouble typing it out. You can copy and paste it. Okay. So once we have those bow tie index files, and now finally we can do our alignments. So on page 43 of the slides, I have the command we're going to use for doing the bow tie alignment. I'm going to switch over to terminal. And so this is the command we're going to use. So we have to do this 4 times for each FASTQ file that we generated. I'm going to start with the stat1 30-minute interferon. Oh, I just realized I sent them to a private chat. 1 second. Hold on, let me send those commands to everybody in the chat. Okay. In case you missed it when I switched the screen. Is that better? Okay. So I'm going to start running the bowtie2 command now. So this is all we have planned for the rest of today. So once you finish doing these bowtie2 alignments, feel free to, you can leave the meeting, you can stick around and ask questions. So I'm going to email the quiz right now. And again, if you are taking the class for credit, it's a requirement. If not, you don't need to take it. So it's through Google Forms, so I need to send those out. And you can have until, I know the workshop's only scheduled to 4:30, so hopefully that gives you about a half an hour to finish it. But if you need to take longer, I'll give you till 5 o'clock because we ran into that problem. So if it says disk quota exceeded, it means that you have run out of room. So hopefully there's files in there that you can remove. As long as you're working in the Flash Scratch, it would be hard to exceed the disk quota because you have 2 terabytes.
Hey, Nathan. How do we know what modules we can load in the Hoffman cluster?
Yeah, so the alignments do take a little bit of time. It might take a half an hour to do all 4. It might, something like that. So each one might take 10 minutes or so. So I'm not sure why that. All right, so I didn't explain. You need to unzip the HG19. So I kind of skipped a couple things, but make sure that, go back to PowerPoint, make sure that you, if you downloaded through the website, to unzip the HG19 zip file and module load Bowtie 2. So Bowtie 2 comes pre-installed on the Hoffman server, but it doesn't, it's not automatically loaded, so you have to load it with this command module load Bowtie 2 in order to run it. And I requested 4 gigs of memory, and it's running fine. I don't think you would need more than that to run this command. So the first one finished for me. What we see is that only 54% of these reads aligned one time to the genome. So those are the reads we're going to keep. This is not a great number. Oh, I need to share the terminal. So, so we have about 11.5 million reads that were kept. I will say that this is a kind of an old data set, even though it is ENCODE. It's almost 10 years old, I think. So it's not the highest quality, and we're not going to have, you know, that many reads, but it'll be helpful downstream to have fewer reads because it'll help make the programs that we use quicker. The more reads you have, the longer these things are going to take. So now I'm going to do the 6-hour alignment. Also, these are very short reads. I think the reads were like 20-something reads. So typically they're at least 50 reads. So I imagine that the shorter reads are probably going to have more alignment issues than if you had longer. So if people are having trouble, weren't able to get the alignment done, and need to stop doing this, I can have them available to download the SAM file, so you guys can continue tomorrow, because it's going to require to have the SAM files for our downstream programs that we're going to use. So let me know if you need me to make these files available to you. Okay, so tomorrow I'll give you guys a command to get the SAM files if you weren't able to do the alignments yourselves. Okay. Second one finished. See a lot of people are leaving. Hopefully that means that people were able to finish doing the alignments. I'm still on the third one, but probably be done soon. And there's just one more. So again, if you feel free to go if you have the alignments done, but if you need help, I'll stick around until my last one finishes and you can ask questions. Otherwise, I'll see you guys tomorrow. My third one just finished. Okay. Since my last alignment finished, it looks like everybody's leaving and there's no questions. I'm going to end the meeting. Thanks, everybody.